Good morning. This is the last recording about photosynthesis, and in this recording, we are going to discuss Kelvin cycle, or the biochemical pathway of the light-independent reactions. As we said before, that photosynthesis consists of light-dependent and light-independent reactions. A Kelvin cycle is called a cycle because it is a biochemical pathway where we start at one compound and proceed to return to the same compound or to regenerate the original compound again. Here, the original compound we start with is the compound known as ribulose biphosphate. So the Ru stands for ribulose, and the B stands for biphosphate, and the P stands for phosphate. So it is ribulose 1,5-biphosphate. It is a 5-carbon compound. So it is an organic molecule that has a carbon skeleton that consists of 5 carbon compounds, 5 carbon atoms, and at position number 1 and position number 5, we have 2 phosphate groups that are attached. This is the original compound we start with, and that's why you have here step 1. And in step 1, there will be a molecule of carbon dioxide that will start the reaction by reacting with ribulose biphosphate. Actually, we will discuss the overall reaction of Kelvin cycle. You don't have to know the minute details of Kelvin cycle, and we are going to explain Kelvin cycle starting with 6 molecules of carbon dioxide and 6 molecules of ribulose 5-phosphate in order to produce 1 molecule of glucose. So the overall reaction of Kelvin cycle is that you start by 6 molecules of carbon dioxide and they react with 6 molecules of ribulose biphosphate in order to form only 1 molecule of the monosaccharide glucose, which is C6H12O6. And in order to do that, you need 18 molecules of ATP. 12 of them are consumed in this step, and 6 are consumed in the last step that will regenerate ribulose biphosphate, meaning that in this reaction, we are consuming 18 molecules of ATP and converting them into 18 molecules of ADP. When we say consuming, we mean that we are hydrolyzing ATP and breaking it down to give rise to ATP. I'm sorry, there was an internet issue. I got disconnected all of a sudden. So we were saying that we are consuming 18 molecules of ATP, meaning that we are converting them into 18 molecules of ADP and generating energy. And that energy will finally be stored. What are we going to do with that energy? We are going to store it in the bonds of glucose. That's why glucose is, first of all, a macro molecule, meaning that it consists of many monomers, okay, which are actually, it consists of many atoms like carbon, like hydrogen, like oxygen. Glucose itself is a monomer. Glucose itself is a monomer, but actually it is a macromolecule. And it is an organic molecule. Why do we consider it an organic molecule? Because it has carbon and hydrogen, both carbon and hydrogen. And at the same time, It is a macromolecule because the carbon skeleton consists of 6 carbon atoms. So all these bonds in between the carbon atoms and in between carbon and hydrogen will store potential chemical energy. On the other hand, what do we require else in order to finalize Kelvin cycle, or in order for Kelvin cycle to proceed? We need 12 reduced NADP to be converted into 12 NADP. If 12 reduced NADP are converted into 12 NADP, this means that they have lost their hydrogen, and that hydrogen will be used in order to reduce carbon dioxide. So if we are asked about the overall reaction of carbon dioxide, of Kelvin cycle, so the overall reaction of the Kelvin cycle, overall reaction of the Kelvin cycle is reduction of carbon dioxide, reduction of carbon dioxide. We reduce carbon dioxide into a monosaccharide. And as we said before, in that chemical pathway, which is Kelvin cycle, where we start at ribulose biphosphate and then we proceed and continue the chemical pathway until we regenerate ribulose biphosphate once again. This is a cyclic chemical pathway. And in order for that reduction of carbon dioxide into a monosaccharide to proceed, how many molecules of ATP do we consume? We consume 18 ATP molecules, and we consume 12 reduced NADP molecules. So the amount of, or the number of molecules that are consumed in Kelvin cycle to form one molecule of a monosaccharide is more than the amount of reduced NADP that is consumed, and that's why every now and then we have to refer to the cyclic photophosphorylation. What is the process of cyclic photophosphorylation? It is the alternative of linear photophosphorylation that occurs in the light-dependent reaction. So every now and then we have to stop the common linear photophosphorylation that occurs in the light-dependent reactions, and we have to replace it by cyclic photophosphorylation, which produces only ATP, does not produce reduced NADP, and whose aim is not to synthesize glucose, but whose aim neither to generate reduced NADP, but it has only one aim, to produce ATP and therefore to replenish the stores of ATP. Okay? Now, it is also worth saying that in order for one molecule of glucose to be formed at the end of Kelvin cycle needs 6 turns. Why does it need 6 turns? Because every turn of Kelvin cycle will take only one molecule of carbon dioxide with one molecule of ribulose biphosphate. And it does not produce glucose unless this is repeated 6 times. Why? Because when you start the reaction by one molecule of carbon dioxide with one molecule of ribulose biphosphate, you have only one carbon atom. And glucose is a hexose, meaning that glucose needs or is formed of 6 carbon atoms, C6H12O6, and therefore we need the cycle to turn 6 times. Of course, there are many details in Kelvin cycle. We are just going to go over the steps rapidly in order to understand what's written here, but you needn't memorize them. For example, if we take the overall reaction, as we said, 6 carbon dioxide and 6 ribulose biphosphate. Let's count the number of carbon atoms. 6 by 1, here we have 1 carbon atom. Here we have 1 carbon atom in carbon dioxide, so we have 6 carbon atoms. And then here ribulose 1,5-biphosphate consists of 5 carbon atoms. If you have 6 of that molecule that consists of 5 carbon atoms, then you will have 6 by 5 carbon atoms, meaning that you will have 30 carbon atoms, 30 and 6. So the overall you started with is 36 carbon atoms. So you wouldn't be amazed if you find that you will have 12 molecules. The number to the left indicates the number of molecules you get, 12 molecules of a 3 carbon atom. How is this 3 carbon atom called? It is called 3-phosphoglyceric acid. Okay, 3-phosphoglyceric acid. Glyceric acid. So the 3 stands for the position of the phosphate. When you write a number like this and a dash after it, and you have a phosphate group in that compound, then the 3 indicates the position of the phosphate group. What does this mean? This means that you have glyceric acid that consists of 3 carbon atoms, and at the position number 3, 1, 2, 3, you will have a phosphate group. So how many molecules of that 3-phosphoglyceric acid do we have? 12. Does it make sense? Yes, it does, because phosphoglyceric acid is formed of 3 carbon atoms. When you have a compound that consists of 3 carbon atoms and you have 12 molecules of it, 12 by 3 gives rise to 36 carbon atoms, which is exactly the amount of carbon atoms we started with when we allowed 6 carbon dioxide molecules to combine with 6 ribulose biphosphate molecules. It's true that it was done over 6 cycles, but to make things easier, because it would be absolutely difficult to understand how it occurs cycle per cycle, and that's why, to make your life easier, we study the process as a whole. We study the overall result of the 6 cycles as a whole. And then what happened here between this compound, 12,3-phosphoglyceric acid, and another compound? Here we have 12, which means 12 molecules. This is the number of molecules. And here we have another compound, which is called glyceraldehyde 3-phosphate. Sometimes, instead of putting the position of the phosphate group to the left and saying 3 glyceraldehyde phosphate, we say glyceraldehyde 3-phosphate. It's the same thing. Whenever you have the dashes here, or whenever you have a number inserted inside the acronym, this indicates the position of the phosphate group. So you have a glyceraldehyde molecule that has a phosphate group. But what's the difference between that compound, which is glyceraldehyde 3-phosphate, and the compound above, which is 3-phosphoglyceric acid? What's the difference between that one, 3-phosphoglyceric acid above, and glyceraldehyde 3-phosphate? Both are made of 3 carbon atoms. Both have a phosphate group at position number 3. You can understand the difference when you see the changes that occurred here. Here we have we have gained energy from the hydrolysis of 12 ATP molecules into 12 ATP molecules into 12 ADP molecules. So there is more energy that has been gained here from 3-phosphoglyceric acid to glyceraldehyde 3-phosphate, and we have gained hydrogen atoms because we have 12 reduced NADP that were converted into 12 NADP. And notice that we write NADPH, but actually NADP is carrying 2 hydrogen atoms. So the 12 reduced NADP, when converted into 12 NADP, will lose 24 hydrogen atoms, and the electrons that are contained in those hydrogen atoms are high energy electrons. So actually, in order to be converted from 3-phosphoglyceric acid to glyceraldehyde 3-phosphate, this is a process of reduction. And then at the end of the day, we have this glyceraldehyde 3-phosphate that is released from the Calvin cycle, and we obtain one molecule of glucose. And this is exactly what happens in photosynthesis. We usually say that we have 6 carbon dioxide plus 6 molecules of water, and we get C6H12O6. So actually we use water and we use carbon. Of course, there is no balance in the reaction because I'm supposed to write here 12H2O and then 6H2O at the end. But the overall is 6 carbon dioxide will give rise to C6H12O6. Okay. This is the overall. I think it's balanced because we deducted the 6 waters from both sides. So we have 6 carbon here, and then we have 12 hydrogen and 6 O. Here we have the 6 O, and then we have this extra oxygen, yes, because we have glucose and 6 molecules of oxygen. Yes, now the reaction is balanced. I forgot to write the oxygen. I'm sorry. So we have, this is the overall reaction of photosynthesis. 6 carbon dioxide plus 6 water gives glucose and oxygen. Sometimes we write 12 H2O and then we write 6 water here, but this is the simplest form. So from every 6 molecules of carbon dioxide, you have only 1 molecule of glucose that is formed. How does this happen? Let's first erase all the editing on the screen. The 12 glyceraldehyde 3-phosphate molecules will just give rise to how many glyceride 3-phosphate? 2 only. What about the 10? And if you remove 2 glyceraldehyde 3-phosphate molecules from 12 glyceraldehyde 3-phosphate molecules, you would get the remainder. The remainder is 10 molecules of glyceraldehyde 3-phosphate. Where are they on the Calvin cycle? This is one question. And what are we going to do with those 2 glyceraldehyde 3-phosphate? Actually, as we said here, glyceraldehyde 3-phosphate consists of 3 carbon atoms. So 3 and 3, because we have 2 of them, 3 and 3 will form 6. So this is 1 molecule of glyceraldehyde 3-phosphate and then another molecule of glyceraldehyde 3-phosphate, and they will form the hexose glucose at the end of the day. So now we understood the fate of those 2 glyceraldehyde 3-phosphate molecules. What about the remainder? Because we had 12 and there was nothing here. There was no more reduction, there was no more addition of hydrogen, only we had the name of the enzyme Rubisco. What did Rubisco catalyze? This is an enzyme protein, of course, and it is a famous enzyme protein. It is the most abundant enzyme on Earth because it is present in all plants that photosynthesize, or in all autotrophic organisms, actually. And it represents 30% of the proteins that are present in the plants. What did Rubisco catalyze? Rubisco catalyzed the separation of those 2 molecules of glyceraldehyde 3-phosphate that will form together 1 molecule of glucose, and the release of those 10 glyceraldehyde 3-phosphate. Of course, as you know, glyceraldehyde 3-phosphate consists of 3 carbon atoms. So if you have 10 molecules of glyceraldehyde 3-phosphate, you would have 10 by 3, meaning 30 glyceraldehyde 3-phosphate. What would happen here? We would need some more energy by hydrolysis of ATP into ADP, and then there will be rearrangement of those 10 glyceraldehyde 3-phosphate. So actually here, what will be done is rearrangement of the carbon atoms, meaning that instead of having 10 molecules, 10 here refers to the number of molecules, 10 molecules of glyceraldehyde 3-phosphate, we would have 6 molecules of ribulose biphosphate. 6 by 5, 30, 10 by 3, 30. So all what happened is rearrangement of the carbon atoms. I hope this overall explanation of Calvin cycle was useful, and we are going to proceed. Here, of course, you have the same explanation, so I wouldn't repeat it. Just like enjoy your time trying to do the calculations and confirm that they are correct. As we said before, the overall reaction that occurs is reduction reaction. Why? Because carbon is gaining hydrogen. We also said that Calvin cycle is a cyclic process, and that at the end of the day, we produce a 3-carbon sugar, which is three. We said when we were explaining Calvin cycle, we said glyceraldehyde 3-phosphate. But sometimes, in order to increase the confusion, we sometimes call it, as we said, we can put the 3 to the left, we call it 3-phosphoglyceraldehyde. Some teachers say pigal, but I don't like to say this. I'd like rather to say 3-phosphoglyceraldehyde. And by the way, glyceric acid ends by the carboxylic group, whereas glyceraldehyde ends by the aldehydic group, and the relationship between the two is reduction, but you don't have to know the process of reduction. Okay, here it is worth mentioning that before the formation of phosphoglyceric acid, there is here an unstable intermediate, and that unstable intermediate is not seen on the Calvin cycle. It is made of 6 carbon atoms, but it's not seen because it's unstable. It doesn't stay for long, and it is immediately broken down into 2 molecules of 3-phosphoglyceric acid, which is this one, and that's why we don't write it on the cycle. It is also worth saying that Calvin cycle has to be driven by the products of the light reaction, meaning that in order for Calvin cycle to proceed, we require ATP and we require reduced NADP, and these are the end products. These are the end products of light-dependent reactions, and that's why we say that in order. One second, please. Okay. I was just making sure that we are recording. In order for Calvin cycle to proceed, we have this has to be preceded by light-dependent reactions. Light-dependent reactions. This is very important to know, okay? And now we are going to talk about a very important problem that occurs in photosynthesis, which is photorespiration. Here, the main enzyme, as we said, is Rubisco, and Rubisco stands for ribulose biphosphate. So the RU comes from here, carboxylase. Why? Because the overall reaction of Calvin cycle starts, or which is reduction of carbon dioxide, starts by what? Starts by addition of carbon dioxide to ribulose biphosphate. This is not the overall reaction. The overall reaction is a fixation of carbon dioxide, which occurs through reduction of carbon dioxide to convert it from the gaseous state into the solid, stable state, which is the monosaccharide. This is carbon dioxide fixation. This is the overall reaction. But the beginning of Calvin cycle occurs through carboxylation of ribulose biphosphate, or addition of carbon dioxide to ribulose biphosphate. So that's why Rubisco is an acronym that stands for ribulose biphosphate carboxylase. Rubisco has a major flaw. Although Rubisco is a great enzyme that does a great job, yet Rubisco has a flaw. What is that flaw? That flaw is whenever oxygen is abundant, and not only that oxygen is abundant, but that the ratio of oxygen to carbon dioxide increases too much. Usually we have a small amount of carbon dioxide. You know that oxygen forms like 70% of the gases in the atmosphere. Carbon dioxide forms a very small percentage. I don't remember. It might be 0.3%, but it's the ratio is essentially a very great ratio. However, this ratio should be very balanced, meaning that if there is an increase in oxygen in relation to carbon dioxide, Rubisco does a major risky steps, or a step that might appear harmful to the plant, which is fixation of oxygen instead of carbon dioxide. How come that Rubisco can do this? Carbon dioxide is the substrate, the specific substrate of Rubisco, but actually oxygen can also be a substrate for Rubisco. So Rubisco cannot differentiate between the two. Rubisco can fix oxygen instead of carbon dioxide. But unfortunately, when Rubisco fixes oxygen instead of carbon dioxide, it looks like respiration. Fixation of oxygen will result in oxidation finally. Why Rubisco? Why Rubisco Rubisco is combining with oxygen, this definitely is for the purpose of making ribulose biphosphate here react with oxygen instead of reacting with carbon dioxide. So the whole process will be different, because we would have oxidation of carbon dioxide. If you fix carbon dioxide, you have carboxylation of ribulose biphosphate. But if you fix oxygen, you would have oxidation instead of carboxylation, oxidation. So this is very similar to respiration, but the process is that you neither get benefit from this nor from that, meaning that when Rubisco fixes carbon dioxide, you get glucose, which is the monosaccharide. This is when we fix carbon dioxide in photosynthesis. In respiration, when we utilize oxygen at the end of the electron transport chain, we produce 38 molecules of ATPs. So in photosynthesis, we obtain glucose when we fix carbon dioxide. In cellular respiration, we obtain 38 molecules. But here in this process, that is called photorespiration. Photo because it occurs after the light-dependent reactions using Rubisco, which is one of the enzymes of photosynthesis, and respiration because it mimics respiration by combining with oxygen at the end of the day. So in this process, photorespiration, you neither obtain the sugar like in photosynthesis, nor obtain ATP like in respiration. So photorespiration seems to be a very wasteful process, because it decreases sugar synthesis. As you see here, it decreases sugar synthesis and it wastes energy. And some references say that Rubisco is committing a molecular faux pas. Faux pas means incorrect step in French. So actually, I believe that it can't be a faux pas. This is something that occurs in the plants, and the plants are surviving despite the occurrence of photorespiration. So it can't be a faux pas, as they say, or a flaw, but it should have certain wisdom behind. Some scientists say that it might be a vestige of the early life on Earth, when oxygen was not that abundant, and therefore the fact that Rubisco can fix both carbon dioxide and oxygen did not cause a serious problem at that time during early life on Earth, because during early life on Earth there was no oxygen, and therefore there wasn't any risk for Rubisco to fix oxygen. Okay, Of course, when photorespiration occurred, this results in the fact that this first step of photosynthesis, which is fixation of carbon dioxide using ribulose biphosphate, would be less efficient. Why would it be less efficient? Why don't we say, for example, that it stops or it is not efficient at all? Why do we say less efficient? Because actually there would be like a competition between oxygen and carbon dioxide, meaning that some molecules of carbon dioxide will be fixed by some molecules of Rubisco as usual and will react with ribulose biphosphate to give 3-phosphoglyceric acid, but some molecules will not be fixed and will be wasted. So in photorespiration, we said it before that it is wasteful because we produce less sugar and we don't produce any ATP, and we are wasting the energy of the cell without forming glucose molecules. Here in photorespiration, what happens to the molecules of carbon dioxide that some of them are fixed as usual into a sugar, whereas some others are lost. Some others are lost and are not fixed as usual. Of course, this is waste of energy, so the plant starts to correct this because you are not forming sugar, you are not forming ATP, and this is the first flaw, and this is the second flaw, and meanwhile you are consuming ATP molecules. So how does the plant correct this? The plant tries to correct this through two organelles. You don't have to know the biochemical pathways of correcting photorespiration, but at least you should know that there are two organelles that share in doing this. The first organelle is the peroxisomes. You remember the peroxisomes that here will try to correct photorespiration, and we will see how. And the second organelle that will try to correct photorespiration is the mitochondria. Through those two they are sequential, meaning that the peroxisomes will act first, and then they will be followed by the mitochondria. We will succeed in saving 3/4 of the carbon atoms and restoring those carbon atoms or retrieving them back to form glucose, but actually 1/4 will be lost. So it is a 74 correction, not 75, percent correction of the flaw, but not 100%. Some scientists find that there are some benefits to photorespiration, like, for example, that some of the intermediates in photorespiration can help immune defenses in the cell. The products that resulted from oxidation instead of carboxylation can produce atomic oxygen and can kill any intruders or any pathogens. Some of them say that they help maintain the redox balance in the cells, meaning that instead of having too much reduction and a defect in oxidized products, this keeps the redox balance. Or they might help photoprotection, meaning that they might prevent light-induced damage. You know that when you're exposed to light, you're exposed to ionizing energy, and that ionizing energy makes you lose electrons, and the loss of electrons is oxidation. And those substances, like the pigments that have been oxidized, can attract oxygen can attract electrons not only from water as they should be, but from other compounds and result in free radicals and oxidation in the cell. And that mainly occurs when we have fluctuating light, sometimes too intense light, that results in a heavy oxidation of the chlorophyll molecule. But all these are not yet proved and they're still under research. Here, of course, it shows you, of course, you don't have to know the diagram, but you have to understand the whole idea. This shows you photorespiration, how photorespiration occurs and how photorespiration is harmful to the cell. And at the same time, it shows you the correction, how the two organelles, peroxisomes and mitochondria, correct photorespiration. And this is one of the functions that should be added to mitochondria. Of course, we know that mitochondria are the energy or the powerhouse of the cell. They are concerned with the production of energy. We also saw that the mitochondria carry some mitochondrial genes that are specific to them and that are responsible for production of energy in the cell, and that those mitochondrial genes are inherited from the mom only. We also saw that the mitochondria can initiate intrinsic apoptosis. They can initiate intrinsic apoptosis. And now we can see that the mitochondria correct photorespiration. Of course, as we said, the correction is only 75% efficient. And here we see how Rubisco can fix oxygen by allowing oxygen to combine with ribulose 1,5 biphosphate. What would be the resultants? The resultants would be 3 molecules of phosphoglyceric acid as usual and 2 molecules of phosphoglycolate. What does this indicate? First of all, we were supposed to have, it is 3 phosphoglyceric acid. The 3 indicates the position of the phosphate group, and here the 2 indicates the position of phosphate in phosphoglycolate. It doesn't indicate the number of molecules. But what does this indicate? It indicates that instead of having all the product obtained as 3 phosphoglyceric acid, we have part of the product obtained as 2 phosphoglycolate, which will not be useful. Phosphoglycerate can continue through Calvin cycle in order to give glucose, but phosphoglycolate will not. So what happens that the peroxisomes and the mitochondria try to correct this by trying to convert. You see the arrow, it starts here, and then phosphoglycolate will go into a chemical pathway, okay, in order to be converted into phosphoglyceric acid. But during that process, we require oxygen, and there is carbon dioxide that is released. And that is what we say, and it's wasteful. And 1/4 of the carbon dioxide molecules are being wasted and cannot rejoin Calvin cycle. So during the process of correction, carbon dioxide is lost. Carbon dioxide cannot be retrieved. And this means one simple thing, that carbon dioxide, this 25%, cannot rejoin Calvin cycle, and therefore cannot be reduced into glucose, Some references say that in this way, photorespiration steals some of the carbon dioxide, or that some of the carbon dioxide is stolen through the process of photorespiration, but I don't like using this term because I believe that all the biochemical reactions that occur in the body, whether we understand why they occur or not, since they occur in a living organism and since it doesn't interfere with the life of the living organism, then it is a useful product. And here you can see how Rubisco loves both. Rubisco loves carbon dioxide and oxygen. Rubisco fixes carbon dioxide and oxygen. Here it's a useful product. That's why Rubisco is smiling. Actually, the diagram shows that Rubisco fixes oxygen but doesn't like it, because Rubisco is sad having committed such a faux pas or a flaw. So here it's smiling because the process is fruitful and produces sugar, and here it's frowning and sad because the process is not fruitful. We do not produce a sugar. Where is the sugar? If we don't correct photorespiration, we wouldn't obtain a sugar, although we are consuming energy and we are consuming molecules of reduced NADP, and we are wasting carbon dioxide. So it's a really big flaw, but the plants are adapted to convert it or to correct it. Okay, now I've added a few slides only to show you. This slide is important because it shows you why does Rubisco do this. We said that Rubisco does this because sometimes there is an elevation in the oxygen to carbon dioxide ratio. Why would be this elevation, or why would this elevation in the ratio occur in hot, dry weather? What's the relationship between hot, dry weather and accumulation of oxygen? Because usually the plants, as you know, have stoma or stomata on their surface, and these are spaces in between the guard cells, as you know, in the epidermis of the cell. In the epidermis of the leaf, and that those stomata are open during daytime to allow for the entry of carbon dioxide for photosynthesis and the release of oxygen that results from photosynthesis. And at the same time, the plant respires all the day. So these stomata allow the opposite also. They allow entry of oxygen for respiration and release of carbon dioxide that results from respiration. So at the end of the day, what would be leaving the cell and what would be entering? Is it carbon dioxide or oxygen? It depends upon which is taking over. If photosynthesis is taking over, if photosynthesis is overriding, for example, during daytime, then carbon dioxide, the net result of these two opposite processes is that carbon dioxide will be taken in and oxygen will be released. At night, when photosynthesis does not occur, so it's only respiration, and the overall or net flow of gases is that oxygen will be taken in and carbon dioxide will be released. Usually, most plants open their stomata during daytime and close their stomata at night. Of course, it's not complete closure of the stomata, but it is partial closure of the stomata at night. But in case of photorespiration, why does it occur? Because when the weather is very hot and very dry, and in the majority of plants, which are called the C3 plants, why are they called the C3 plants? Because the first intermediate compound that is formed is a 3-phosphoglyceric acid, and it is a 3-carbon compound. So we call them C3 plants. These represent the majority of plants, and in these plants, these plants are not adapted to living in hot, dry weather. And if they're not adapted, so what they do is that they close their stomata not only during night, but during daytime. Of course, I'm talking about partial closure. So they close their stomata partially during daytime. Why are they doing this? Because they are in hot, dry weather, and there is the risk of too much evaporation, not only from the stomata, but from the surface of the epidermis in between the stomata. So the plant, the underlying tissues would suffer from dryness. So what they do is they minimize evaporation. This, of course, happens mainly in desert plants, but desert plants, as we will see later on, are well adapted. We're talking now about an ordinary plant that is not adapted and that for a reason or another is unexpectedly exposed to hot, dry weather. When they close, so photosynthesis is going on. Photosynthesis is proceeding during daytime. So carbon dioxide will be taken in through the partially closed stomata. Oxygen will be released, but not released as usual. So what happens is that oxygen will build up inside the air spaces. Oxygen will accumulate inside the air spaces, and that what makes the plants photorespire. Okay, so here we have rubisco. Rubisco, when the weather is fine at low temperatures, it fixes carbon dioxide. So this is the ordinary, this is the norm. When the ratio of oxygen to carbon dioxide is normal and there is no buildup of oxygen, so carbon dioxide is fixed normally through Kelvin cycle. But if we have high temperature, and dry weather, then there is a great possibility of evaporation. So the stomata are closed, and in such a way, oxygen will build up. Oxygen, of course, from photosynthesis will build up inside the cell, and this will lead to photorespiration. Where does this oxygen come from? This oxygen resulting from photosynthesis that continued despite the partial closure of the stomata. So when oxygen builds up, we shift to another process, which is photorespiration. Okay. Okay, all what's on this slide has been written before, so you needn't know all these details, okay? You needn't know how this is done, but I kept it for myself, so you can skip those two slides because they have been explained before, so we would skip this, and you can also skip that. The only thing that is important here is that the two carbons of phosphoglycolate are not important in photosynthesis. They cannot join Kelvin cycle. Phosphoglycolate is the result of the FOPA. Phosphoglycolate should be phosphoglyceric acid. Now, but we said that there is some adaptation to photorespiration, and this is the C4 plants. This occurs in the C4 plants and in the CAM plants. We are going at first to see a table that I've added to the presentation, and that would help you understand the difference between the two. Now, the C3 plants, so this is a new slide. I needn't re-upload the presentation since you will be watching the video. So the C3 plants are not adapted to hot, dry weather. So in hot, dry weather, they close the stomata day and night. Of course, this closure of the stomata is partial, but it has an effect on the plant. Why do they do this? To minimize evaporation. What's the result of this? The buildup of oxygen. And what's the result of buildup of oxygen? It is photorespiration. And as you can read here, C3 plants usually open stomata during daytime to allow carbon dioxide for photosynthesis and close stomata at night to minimize evaporation. And now we have two plants, C4 and CAM, that are adapted. Adapted to what? Adapted to hot, dry weather. And since they are adapted to hot, dry weather, so there is no photosynthesis that occurs. How do C4 plants do this? They keep the stomata open during daytime, meaning that they are not forced to open the stomata during daytime, and I would add despite the hot, dry weather. They have enough courage to keep the stomata during daytime open despite the hot, dry weather. Why? Because they are not afraid that, or they are not worried about the fixation of oxygen by Rubisco. Why? Because in this way, when they open during daytime, first of all, there is no buildup of oxygen. Second, they can fix carbon dioxide in deep cells away from oxygen. So there are two benefits here. The first benefit is they open during daytime, so there is no build up of oxygen. Second, they are more protected by forcing Rubisco to fix carbon dioxide away from oxygen in very deep cells, whereas the CAM plants are well adapted because they close the stomata during daytime and open at night, meaning that they do not close 24 hours over 24 hours as the C3 plants did. The C3 plants closed 24 hours over 24 hours, and this was the result of or this resulted in the buildup of oxygen. Here we open at night. First of all, this is the opposite of what all plants do, opposite of all plants. But why are they doing this? They are doing this opening at night because at night it is less hot and therefore there would be less evaporation. So this is the first benefit they get. The second benefit they get when they open, so the first benefit is they do this opposite to all plants. It is less hot and there is less evaporation, and therefore there is no fear of evaporation. The second benefit is that when they open at night, this means that they open 12 hours over the 24 hours. So there is no buildup of oxygen and there is no photorespiration. This is the most important thing you have to know about the adaptation of the C4 plants and the CAM plants. But how they do this is too many details. I'm just going over in order to just give you an idea about this. And I will tell you what you have to know only concerning these biochemical pathways. Here we see the first alternative pathway, which is the C4 plants. We said that the C4 plants have the privilege of fixing carbon dioxide away from in deep cells, as we said here, and they are not afraid, they're not scared, they keep their stomata open during daytime, meaning that they open their stomata 12 hours over the 24 hours. Why aren't they scared? They aren't scared because, they can fix carbon dioxide in deep cells. Why are they? These are the deep cells. So here we have two types of cells. And for example, if we look at the epidermal cells, and then here this is the epidermis, and then we have the perpendicular palisade cells, and then we have the mesophyll cells, which are large and irregular, and then we have the bundle sheath cells. The bundle sheath cells, as you see here, so these are the mesophyll cells, and then here you have the bundle sheath cells. As you see, the bundle sheath cells are very deep here, and therefore when carbon dioxide is delivered to Rubisco in the Calvin cycle, there is no risk of photosynthesis. So How can we prevent? First of all, Rubisco is not present here. The enzyme that is present here is not Rubisco, but it is an enzyme that is called PEPC, or phosphoenol pyruvate carboxylase. Phosphoenol pyruvate carboxylase. So it fixes carbon dioxide to a compound other than ribulose biphosphate. So neither ribulose biphosphate nor Rubisco are present here. We fix carbon dioxide to phosphoenol pyruvic acid, and we get oxaloacetic acid. So the first intermediate here is a C4 compound, and that's why we call those plants C4 plants. The ordinary plants, as we said, gave phosphoglyceric acid, which was a 3-carbon compound, and that's why they're called C3 plants. And then oxaloacetate is converted into aspartate, and then aspartate is pumped actively into the cell to give rise to oxaloacetate, and carbon dioxide is released once again to join Calvin cycle, and the phosphoenol pyruvate is returned. This is the overall process of the C4 plants. In the CAM plants, the process is very similar to the C3 plants. All what they do is that they photosynthesize at night. So here, the main characteristic of the CAM plants is that the stomata open at night. This is, of course, a major privilege. Why? Because when the sunlight, touch or hits the plants and the light reactions occur, what happens in the Calvin cycle is that they actually, sorry, I'm sorry, I started the wrong way. The stomata are open at night, so carbon, we have to start here. So we have to start here, okay? This flaw usually occurs at the end of the presentation when I give too many details and I get tired. So here we start here, and carbon dioxide enters the cell. But carbon dioxide, what happens is that carbon dioxide during night is converted into malic acid or malate. And then malic acid during daytime will be joining Calvin cycle when the stomata are partially closed. So the CAM plants, they open their stomata mainly at night, and they partially close their stomata during daytime. What do they benefit when they partially close their stomata at, what do they benefit when they open their stomata at night? As we said, they are not afraid or worried about evaporation because the weather is better at night. So they take in carbon dioxide, okay? But what they do with that carbon dioxide is to fix it into a 4-carbon compound that is malic acid. And that malic acid is stored into a vacuole deeply in the plant in general, in the palisade cell, not deeply. Palisade cells are superficial cells. And it stays all night, the plant stays all night fixing carbon dioxide and converting it into malic acid that is stored inside vacuoles inside the palisade cell. And then during daytime, when the stomata are partially closed and when light starts to reach the plant, and why are we saying this? Because we would have light reactions that would generate reduced NADP and ATP. Light reactions would proceed. Now it's high time that we release malic acid from the vacuoles into the cytosol, then to Calvin cycle, where it will release carbon dioxide again and start Calvin cycle. So the overall process gives you the impression that we are storing carbon dioxide at night. We are storing carbon dioxide at night in the form of, this is the overall process, we are storing carbon dioxide at night in the form of malic acid. And then after having stored it in the form of malic acid in vacuoles, then we will release it during daytime. We will release carbon dioxide during daytime, during daytime to the cytosol first. Okay, we will release carbon dioxide during daytime, but it is only released when inside the chloroplast, when malic acid reaches the chloroplast. So we will release carbon dioxide during daytime into the chloroplast. So you can see that carbon dioxide cannot have a journey in the cytosol. It is transported inside, hidden inside the malate molecule and released only in the chloroplast. Of course, as I said, the last 10 minutes of the presentation where I have explained the adaptations of the C4 plants and the CAM plants, they are important, but you just have to know the overall details. Thank you, guys. I know it was a bit lengthy, but I was keen on giving you all the necessary details so that you can have all the background that would help you answer any question related to photosynthesis. Thank you so much.